Hello everyone. In this tutorial, I will show you how to build a factory for creating fully working Python applications tailored for medical research from the click of a button. This is a project that was done here at Harvard in the context of an advanced Python course. So in the next few slides, I'll tell you guys about the problem I wanted to address. I'll go over the objectives of this project. I'll explain the approach I have chosen and the exact tools I have used to implement that approach. And finally, I'll go over a short demonstration of the tool. In recent years, there has been a growing awareness and concern that preclinical studies are not always reproducible. And this is really a big problem in medical research because clinical trials in humans are built on studies that have demonstrated a particular effect. So if you're administering drugs to patients based on unreproducible results, you not only put their lives at risk, but you also waste a lot of resources in doing so. So why are there so many studies that are not reproducible? So one possible explanation is that there's a lot of competition and pressure for scientists to secure funding to continue their research. And because people often rely on publications to measure success, it has given rise to a publish or perish sort of culture in many places. For this project, I'll focus on more specific issues within neuroscience research. Neuroscientists use different models to study the nervous system, ranging from individual cells to entire organisms or animals. Each of these models requires different equipment, and each of these devices generate their own type of data. And more often than not, the machines do not come with satisfying software to process that data, so scientists end up making their own tools with limited programming experience. And this can lead to poorly designed software where even two scientists making a tool for the same purpose might get different results. This might underlie some of the reasons why science is not always reproducible. The objective of my project was to give scientists the ability to create Python applications that generate reproducible results. I wanted to provide them with a reproducible workflow that enforces Python's best practices and also allow packaging and distribution of the app to facilitate sharing across scientists. So here's an overview of the approach I have chosen to tackle this problem. First, I wanted to have a project template to ensure that scientists would start with a reproducible scaffold that they can then build upon. Then I wanted to design one Python module for each device or category of experiments. For example, if you're looking at cells, you might be using different types of microscopes, and I wanted to provide the ability to deal with that within a microscopy module. Similarly, sequencing data might refer to DNA or RNA segments, and I wanted a sequencing package to provide code to deal with that. I also wanted a utilities package to provide general operations that might be useful across different devices. For instance, a lot of the data coming out of these devices is either images or video files, and I wanted the utilities package to provide the ability to quickly convert between these different data types and also ensure that the mechanism for doing so would be standardized across scientists using this platform. I also wanted to provide a skeleton graphical user interface for people interested in interacting with their data or to share with colleagues that have no programming experience. And the idea is that all these modules would be optional user-selected features of the overall template. To complement this interface I was talking about, I also wanted to provide packaging scripts to bundle the app into a single executable file for easy distribution. I also wanted to provide an environment that is both tested and has the framework for testing custom-made features. Lastly, I also wanted to enforce some of Python's best coding practices using tools like Flake 8 or Black, among others. Now let's go over the actual implementation of this approach. The templating system was implemented using Cookie Cutter because it really provides an easy way to create identical Python package projects. Within that template, the Docker files necessary to deploy and run an application within a Docker container is provided. This provides a fully contained and reproducible environment. pipenv is then used to keep track of a consistent set of Python packages necessary for the application, so pipenv provides the Python environment here. Inside this Python environment, I implemented the Python modules I described a minute ago. Finally, I implemented unit tests using PyTest. I also have Flake 8 running simultaneously to check that new code is consistent with Python's coding conventions. And PyInstaller scripts are also provided here to bundle the app into a standalone executable file. So let me frame all that in terms of reproducibility so that we understand how this workflow can help make science more reproducible. So the cookie cutter template provides a reproducible way of creating new projects. The Docker container provides a reproducible way of running things. pipenv provides a reproducible Python environment. Flake 8 provides a reproducible way of ensuring code is consistent with Python's conventions. PyTest provides a reproducible way of running tests. PyInstaller provides a reproducible way of packaging the app for distribution. The Python modules themselves provide a reproducible way of performing computations and manipulating data from different devices used in research. Now let me explain how advanced Python constructs were leveraged to implement the Python modules. Inheritance was used in the context of animal behavior analysis. Ethovision is a commercial software that allows recording and tracking of rodents in a given enclosure, but the system can be used with any enclosure. So one scientist might use it with a water maze, where a mouse is free to swim around. Another scientist might use it with a place preference setup, where a mouse can choose to spend time in a light chamber or a dark chamber. So inheritance was used here to provide a parent class, the Ethovision class, for preprocessing the output of the Ethovision software. The children classes include a common preprocessing step inherited from that parent class, but additionally provide analysis that is specific to the enclosure chosen by the scientists. Decorators were also used to implement some of the functions provided in the utilities module. Specifically, parameterized decorators that act as decorator factories were used to allow conversion and saving of the output of a function. Here, the save video decorator intercepts the output of a function and saves it to a video file with a specified frame rate. That might be useful if you want to convert a series of microscopy images into a time-lapse video, for example. The Save Images decorator does the opposite and converts a video output into a series of images. These decorators can also be useful when you're applying many functions to transform your images and you want to save the intermediary steps for debugging purposes. Now let's talk about testing. Testing is an important part of doing rigorous and reproducible science, and I spent a good amount of time implementing tests for everything that is provided in this project. PyTest is used to run the tests on the individual Python modules, and it provides a nice line and branch coverage report. Travis is used to provide continuous integration testing as well. The testing phase really consists of two stages. First, the templating system itself is tested to ensure it generates the right project structure. Then, in the second stage, an instance of that template is rendered and all of its modules are thoroughly tested. Now let's go over a short demonstration. For this demo, let's assume I'm a scientist using this particular microscope to visualize a population of neurons. The microscope allows us to watch these recordings and also export the brightness of individual cells at each time point. However, the output isn't really clear and doesn't give us any comparable metric of cellular activity. So to address this, we will use this project to generate an app that can take the output from the microscope and turn it into useful and readable measurements of activity. Before we begin, make sure you have Python 3.7 as well as pipenv installed on your machine. Then copy the directory from GitHub. Once you've copied the directory on your local machine, you should see a directory structure that looks like the one here, where you have the hooks folder, which contains script to run before and after generating a template. Template cleanup is done in the post script here. The test folder contains unit tests to test the templating system itself. The folder here is what gets rendered as new and independent projects. Inside of this, you have the Docker files necessary to run a new app inside of a Docker container. You also have bundling script that allows you to package your app using PyInstaller. You also have pip files to control the Python environment inside of which you're going to be running your app. Resources folder contain the icons that are used when making an app with PyInstaller. The folder here contains the main features that are provided that you can use as part of your app. Ethovision is used for animal behavior. Microscopy is used for processing of microscopy data. Sequencing is used for processing of RNA, DNA sequencing data. The GUI is the graphical user interface that we're providing. The util package contains general data manipulations like converting between file types, and we can look, for example, at the data converter here. And for instance, we're providing two decorators, one for saving the output of a function as a video and the other to save it as images. Now let's go ahead and create the app we were just talking about. First, browse into the project directory. Then run the following pipenv command to set up your Python development environment. Now activate this environment by running the following pipenv command. You are now ready to create your new app. Move up one directory. Now you can invoke the cookie cutter command on the directory. Here you can answer project-specific questions. For this example, we're just going to use the default settings and press enter for each question. Include GUI, include utils. These provide you with the ability to determine what modules you want to include in your app. The lab project app should now be visible. Open the project directory in the editor of your choice. Here we're using Sublime 2. Now we're going to go ahead and modify the interface file so that we can invoke the IXM file parser, which is what's going to allow us to process the output of the microscope. Here we want to import the IXM file parser from the microscopy package. Then go ahead and browse down to the analyze function and add your code here under do some analysis. First we want to instantiate the parser. Then we want to invoke that parser on the file that is selected by the user on the interface, as you will see shortly. We are now ready to bundle the app and see if we can select the output file from the microscope and turn it into a meaningful output. Go back to the terminal, browse into the project directory, and run the following pyinstaller command to bundle the app in a single standalone executable file. This will create a distribution directory that contains your fully packaged app. Double-click it to launch it. An interface should be open to allow you to select a file, analyze it, and close the interface. Click Select File and select the output file from the microscope. Here it's called neurons.csv. Click Open, click Analyze. At this point, it doesn't tell you that the analysis was completed, but let's go ahead and check if the output file is present in our directory. So as you can see, it created an output folder with our output data, which looks as we expected with easy to read features about each of our neurons. So this is how you can build an app using this project. This tool will help scientists make their data analysis more standardized and help make science more reproducible in general. Thank you for watching.